Hello and welcome. Thank you so much for joining us for what will be a fantastic deep dive into how we are using the latest technologies, including artificial intelligence and stem cell biology, to solve the mysteries of brain disease. Today's webinar will introduce why diseases of the brain have proven so difficult to understand and treat, and how we are using a combination of cutting-edge approaches at the NYSCF Research Institute to truly pave the way for effective new therapies. In today's webinar, we're only able to touch on a small portion of everything that we do at NYSCF. If you're interested in learning more about anything discussed, or if you have a specific disease interest, please reach out via our website or through the chat function. Before we dive in, I want to acknowledge the transformative partnership of the Stavros Niarchos Foundation in making events like today's talk possible and encouraging others to support our efforts. If you would like to make a gift toward our education and outreach programs, all gifts will be generously matched by the Stavros Niarchos Foundation one-to-one. And we really hope that you will consider doubling your impact with a gift today. You can visit the link in our chat for more details. And to all of our visionary partners who are watching today, we're deeply grateful to you. Our research and discoveries continue to be fueled by your philanthropic support. For those of you who are new to NYSCF, we look forward to sharing more of our work with you and hope that you will join us in our mission of accelerating cures for patients everywhere. I'm so glad to have one of our innovators with us today, Dr. Vikram Khurana, a leading physician researcher at Harvard Medical School who focuses primarily on Parkinson's disease, but also other neurological diseases. And Vic actually sees and treats patients every day. The clinician perspective is so important to consider as this research process moves ahead, and we are really thrilled to have Vic's input on tackling the challenge that brain diseases present. And most critically, it is imperative to understand and consider the patient perspective in any research pursuit. We are thrilled to welcome back Leena Panwala, the founder and president of the INAD Cure Foundation, who is unfortunately living the day-to-day experience of patient advocates everywhere as she desperately seeks new treatments for her daughter, Aiyar, who suffers from infantile neuroaxonal dystrophy, INAD as it's referred to, which is a rare and debilitating disorder that attacks the nervous system of children. I am happy to say that Leena is not alone in her search for better options. We are working very closely with Leena and the INAD Cure Foundation towards an actual cure for INAD. And to moderate our discussion, I'm delighted to welcome back the fantastic Dr. Raeka Aiyar, NYSCF's Vice President of Scientific Outreach. Raeka is an expert at translating the complex scientific concepts that we work with every day into understandable and clear outputs for our entire community, including both fellow scientists and people who have never heard of a stem cell before. And as such, we're really lucky to have her with us today. To start off, I'm going to introduce Dr. Daniel Paull, NYSCF Senior Vice President of Discovery and Platform Development. Dan leads our NYSCF Global Stem Cell Array team at the NYSCF Research Institute and has played a key role in spearheading its development. The array is our unique robotic platform technology that allows us to create induced pluripotent stem cells and specific cell types. This allows us to study disease through the lens of what's happening in patient cells at very large scale. Dan spearheads technology innovations that allow us to see this even more clearly, and we'll hear from him today as he discusses NYSCF's latest study about to be published, which successfully applied AI, artificial intelligence techniques, to Parkinson's disease. Dan also leads NYSCF's discovery efforts using the array platform as we have combined it with artificial intelligence, genetic engineering, and more to find better treatments for patients faster. We need all of these technologies to tackle these very tough diseases. Dan is the perfect person to walk us through the approach that we take at NYSCF and to set the stage for our discussion today. So Dan, welcome.
Thank you very much, Susan. Thank you all for joining today. So a brief introduction to some of the technology that's underpinned the latest study that we're about to publish, as was said, as well as touch on sort of what we're doing next with this. And so to start with, just a slide that maybe some of you will have seen before, but an understanding of what is difficult, or at least some of the things that are difficult in finding treatments for diseases. Many drugs that are in development don't ever reach patients. They fail in clinical trials, many times due to a lack of effectiveness or because of safety reasons. And it could just be that in some cases for these trials, we're just giving the drugs, medicines to just the wrong group of people. And so on that front, we have many drugs that do exist. And what we see is that for some patients, they work incredibly well. And for others, they don't, or they induce side effects that make them detrimental to the patient's quality of life. In developing a drug, it is a very long process, averaging maybe over a decade of time. It's very expensive in the billions of dollars. And if many of these are ultimately leading to failure, it can be a very challenging space to work in. One of the other things that makes it difficult is that the way we develop drugs, they're often based on models that don't necessarily capture the intricacies of human health. And so in that, we're often using animals to model these diseases, but even using human cells, they don't always faithfully recapitulate what actually happens inside the human body. So in order to sort of redefine and change how one might think about drug discovery, we think that there are some avenues to go down that can maybe provide new paths forward. And we think in order to do that, it requires the combination of a few different areas. And one of those is robotic infrastructure, advanced imaging techniques that allow new insight and new ways of looking at cells, and combining that with new ways of then analyzing those images. It's very hard as a scientist to look at too many images in one go, but when studies get larger and larger, this is something we want to take advantage of. And so if we combine all of these three things in our latest study, that's exactly what we did. And I'll walk you through how the combination of those has allowed us to unlock new understandings of Parkinson's. So primarily, as Susan mentioned, we have a platform here called the NYSCF Global Stem Cell Array. It is a set of instruments that act in much the same way as human hands do. They can pick things up, as you see here, with a plate, and there are cells growing in here under that liquid that you saw bouncing around. We have robotic arms that can move these things between, say, a microscope, an incubator where the cells live, and ultimately onto the robotic platforms where we can feed the cells and perform our experiments. This platform runs 24 hours a day, 7 days a week. So when we're sleeping, the robots are still running, taking pictures and generating, thousands of data points on a daily basis that we put into our database here. And it's really this scale of being able to use robots like this that's allowed us to build very, very large experiments. Instead of an experiment having the cells from maybe 2, 3, 10, 20 people, we're now at the point where we can conduct experiments that involve many hundreds of different patients' samples. So one of the ways that we've been looking at this is by using artificial intelligence to help us in this process. And so one can imagine that if you have a set of samples from healthy individuals and a set of samples from patients suffering from a given disease, if you take enough pictures and the right kind of pictures of cells from these different individuals, we can perhaps use artificial intelligence to identify features within these images that otherwise are not visible by the human eye, or things that we would overlook, disregard. Again, it's very, very taxing to look through such images. And so we took this approach into Parkinson's disease, and we really set about doing this in as unbiased an approach as we could. And ultimately what we wanted to be able to do was classify any set of images as either coming from cells of a healthy individual or cells of someone suffering from Parkinson's disease. And to do this, we relied on what's called a neural network. And for this, we worked with great collaborators at Google to help us with this work. Sorry. We used our automated pipeline to take millions upon millions of images of cells that look just like this. And importantly here, although stem cells are the majority of the cell type we use, these were done in human skin cells. So a simple skin cell derived from human skin. And we took millions of images and had put them through a process that we know as cell painting. And that painting process is what makes these look so beautiful. These cells have gone through a round of expansion. And in order to be able to have had these cells, we were able to go into our JAX-NYSCF repository here. And this is a large collection of cell lines from many thousands of people. And we were able to take around 50 Parkinson's patients and around 50 healthy controls and use these in our study to ultimately work through this entire workflow. As I mentioned, we took millions of images, just under 6 million in total, and they look a little bit like this. And again, to the human eye, you can see some patches here that maybe have slightly less cells or more cells. That's a given in biology sometimes, but it's hard to find discernible differences that correspond to a given disease. So feeding this into our advanced machine learning models, we again asked it if we saw enough pictures, could the model identify whether any given image it was looking at was from a healthy person or from a person with Parkinson's disease? And ultimately, and what we're about to publish, is that the imaging platform could indeed lend itself to being able to distinguish between these two individuals. And if you want to read more and read into this in a bit more detail, the links to the manuscript itself and some written up reports are available in the chat window. So the next step we're applying at the moment is then, if we can tell the difference between cells that have come from an individual with Parkinson's and an individual who is apparently healthy, can we find a medicine or a drug that can convert or change the appearance of those cells to look healthy again? And perhaps at that level, we've then got something that would be therapeutically useful to patients. We're now using this same platform and expanding it across a number of other areas, including rare diseases such as INAD, as well as things like aging as well. And that's it from me. And now we can open up the chat.
Great, thank you, Dan, for giving us a great preview of the topics that we're going to get into today. It's now my pleasure and honor to be moderating today's discussion, and I'm grateful to this fantastic group of panelists for joining us. We're going to delve into the potential for AI to change the way that we study and treat diseases. So thank you to those of you who have submitted questions in advance, and please add any questions that arise along the way using the Q&A function, and we will do our best to get through as many as possible. I want to start now by bringing on our panelists and asking them to help us start this discussion by highlighting some of the challenges in disease research that we hope that AI can help us to address. So I'm going to start with Vikram Khurana. Vik is the chief of the Division of Movement Disorders at Brigham and Women's Hospital and at Harvard Medical School. Vik, thank you for joining us. You're a physician, as Susan mentioned, who treats Parkinson's and several similar diseases. What can you tell us about the current treatments available for these diseases and the opportunities for improvements? Where are the gaps, and why has it been so hard to find more effective treatments than what we have at our disposal?
Thanks so much, Raeka, and thank you everyone for asking me to participate in this really good discussion, and very timely. That's a complicated question. There's many things that I'd say. So I'm going to frame in a simple way, and then hopefully some of the themes might come up in the rest of the discussion. So the first thing is to say, and I'm going to center this on neurodegenerative diseases, since we started by talking with Parkinson's disease.
Could you explain what a neurodegenerative disease is?
A neurodegenerative disease is one in which there is thought to be correct development of the nervous system, but that at some point in that individual's life, something triggers the dysfunction and the loss of brain cells, neurons, and glial cells, often the supporting cells of neurons in the brain. And that leads to a progressive deficits in neurologic function, and these could relate to our cognition, memory, these could relate to our motor function, and we've all heard of terrible diseases like Alzheimer's, Parkinson's, Lou Gehrig's disease, and that really covers, a set of these diseases, gives you a sense of their the devastating effects that they can have on patients. So that's a neurodegenerative disease. Now what we really, it's not fair to say that we don't treat these diseases. We do treat them, but we treat them symptomatically. We don't alter the course of these diseases. There are many symptoms we can treat, and sometimes that can be effective. For Parkinson's disease, giving a patient dopamine or related drugs can give a fantastic relief of symptoms for years, but it doesn't change the ultimate outcome of the disease. And frequently, our patients will continue relentlessly towards neurological decline. So symptomatic treatments don't change the course, but they can be helpful. And by and large, that's what we have for these diseases. Now, if folks are interested in talking about Alzheimer's disease and aducanumab, we can talk about that. Controversial recent decision by the FDA supports the possibility that an antibody to beta amyloid may be disease modifying, but that's still early days for us to kind of decide on whether that's really the case or not in terms of neurologic function for patients. But by and large, symptomatic treatments. And then, I think it's just going to be important for the discussion in general. There are, let's call them simple monogenic diseases, neurodegenerative diseases caused by a known change in one gene, a one gene mutation, for example. And then there are complex diseases, by which we mean we don't exactly know what the genetic cause is. We make an assumption that it's complex, it's made up of different genetic factors, environmental factors, and most patients with diseases like Parkinson's and Alzheimer's disease are in this category. They have complex disease, and that means we can't quite put the finger on what's caused these diseases. And really, that's kind of, I think, a nice way to bring up the big challenges of degeneration and maybe where AI can be helpful, because ultimately the problem with treating complex degenerative diseases, what most people have, is that patients may not really have exactly the same disease. We may be dealing with disease heterogeneity. We may be dealing with different flavors of disease. And the great tragedy here is that where most of our clinical trials failed, it is possible that subsets of those patients responded beautifully to the agents that were so meticulously put into clinical trial, and yet we would never know, because we clustered all of our patients together and we gave them all a single drug and a placebo, and we were looking for some signal in that noise. And in order to succeed, it means you have to have such significant effect across that population that within the framework of a disease that unfolds for years, that within 6 months or 12 months, you would have such a significant effect in a subset of those patients, that you would see a signal. And that bar is really high. So we're almost setting ourselves up for failure when we do any kind of clinical trial for a neurodegenerative disease, because the challenges are enormous when you might be dealing with patients who have different sets of diseases. And so I'm not going to talk in huge detail. Obviously, everyone else needs to participate in this, but each of the we could break this down into some simple challenges, and we can then talk a little bit about how AI might help. First is you want to be able to diagnose your patient early. We think that early intervention may be important for these diseases in order to shift the needle. That means you need early intervention. So that's one big challenge. Imagine if you're enrolling patients with early disease for a clinical trial that lasted a year. If those patients don't progress appreciably in that year, you're not going to be able to say that your drug has actually fixed any of those patients, because yes, you got early patients, but they were going to show their symptoms in years, and your drug trial only lasts for 1 year. So
Right.
it's not just the diagnosis, but it's diagnosis of a patient population that is actively progressing but early. Okay, that's really hard. So you need that set of patients. Now you want to follow your patients. You need a combination of clinical assessments, biospecimens, biometrics, bioimaging to follow those patients. That generates enormous amounts of data. Our current way of following patients and seeing if we're making any difference with our drug are very crude, very noisy. I can see a patient in the morning and get a rating scale on their movement disorder that is 10 points different from what I see in the afternoon. So if it changes that much during one day, how are we going to use these noisy scales that we have to follow a patient? And finally, the key, treatment. If we're going to create a disease-modifying treatment and put something into clinical trial, we need treatments that are matched to subtypes of patients. If we're going to say patients have different characteristics, and I want to stress that even in patients that have a single gene mutation that causes one of these diseases, there can be huge differences in the same family of how those patients manifest their diseases. And so that means we need a way to classify our patients. And so using genetics, using the kinds of stem cell tools that we're going to talk about today, these are avenues for trying to subtype our patients and match a therapy for something that we think is important to that patient. And one final point I'll make is that we've been so fixed on genetics, but remember, genetics tells us about someone's risk for the disease. It doesn't necessarily tell us what dictates how they progress with their disease. So if you've got a risk of a disease and now you come to see a neurologist, the factors that are driving the progression of your disease may be different to the factors that are driving your risk of the disease. And you need to be designing a drug that looks at the progression, not the risk. And those two may be related, but they may not be. So that's just a broad introduction to maybe the big challenges that we face at different levels. And hopefully our discussion can flesh out how some of the beautiful work that NYSCF and others are doing might help address some of these challenges.
Thank you so much, Vic. Really, really great points to think about, and we'll definitely come back to the idea of subtyping patients and subgrouping patients for precision medicine as we go. I now want to bring on our next panelist, Leena Panwala, who's the president and co-founder of the INAD Cure Foundation. As Susan mentioned, you're the mother of a patient with INAD, which has led you on this disease advocacy journey where you've done extraordinary work to advance research and treatments for INAD patients. Can you tell us what this experience has been like for you and what kinds of challenges you've encountered along the way?
Yes, Raeka, thank you for that introduction and for asking me to participate. And as Susan mentioned, INAD is a neurodegenerative condition. It stands for infantile neuroaxonal dystrophy, and it starts in very young children, from anywhere from between 6 months to 3 years of age. And as Vic just kind of discussed, it does present differently in each child. We've had siblings that have the same mutation, but go on to have different sort of symptoms and effects of the mutation. And it's very interesting and fascinating, and definitely, some of the precision medicine that comes out of this AI, work that's happening could ultimately be more kind of effective and beneficial in treatment options for some of these children. So when my daughter was diagnosed, I had to accept that there was no treatment for this disease. It wasn't on anyone's radar, and it's considered ultra rare. So with a patient population, a global patient population of 150 children, resources really weren't allocated to try to learn more about the disease or how to effectively treat it, because, our numbers are just so small. So the team at NYSCF joined me in my quest for searching for treatment options for INAD. And, we worked together to collect skin and blood samples from multiple children affected by INAD, and we created fibroblasts and iPSCs so we could use these disease cells for all types of research, including drug discovery, repurposing, and with the hope that advancing our understanding of INAD could lead to advancing our understanding for other diseases that affect the brain. And so, we kind of been working through all the steps involved of really trying to get closer and closer to some of these treatment options for these children.
Thank you, Leena. And we'll definitely come back to more of that work in detail. I'm excited to talk about that. I'll now move on to Dan. So Dan, you mentioned during your slides that drug discovery is in general a really challenging process, and it's prone to a lot of failures. Can you tell us a little bit more about the traditional ways that drugs are discovered, from kind of all the way from the research side where we work to clinical trials, and where might be those challenges and opportunities for improvement?
Yeah, really there are hurdles across the whole spectrum of drug discovery. Thinking about basic research at the beginning, just having a model that recapitulates the disease at a given point in time is a challenge. Thinking about more complex diseases such as Parkinson's and Alzheimer's, although it has been maybe easier to get access to samples from those patients, I think Vic speaks to the heterogeneity, the differences that exist from patient to patient, that in itself is, although there is more material available for researchers to use in research, it is confounded by that difficulty. The opposite is true in something like INAD, where perhaps the disease is sometimes more homogeneous, and we know why the disease is happening. There's a specific defect in the genome that causes it. Access for researchers to get hold of that material is very, very difficult. And so studying the relevancy of a drug on a given set of cells has been enormously challenging. And that's why working with Leena and other foundations has been such a driver for us to make those available. I think beyond that, knowing what you're looking for becomes the second problem in this. And so, you can take agnostic approaches and screen, millions of molecules, but unless you have something to measure on the other side of that, something to quantify that you can say, hey, this drug is changing this, having that in the first place is itself a challenge. And so having good, what we call assays to measure a phenotype or measure an outcome of how a drug might be changing something is very challenging. It gets more complex as you go further down the pipeline. We move into things like animal models. Again, they are useful. Some of them are great. Some of them are at best not so great. And so their utility becomes a big challenge. If something's not working in an animal model, does that mean it's not going to work in a human? Not necessarily, but that's sort of the gatekeeper to moving forward sometimes. Vic mentioned it, if you do get toward clinical trial, just the patients that are enrolled in that trial, are they the right group? Are they the right subset of patients that you want to be taking a particular medicine? So if you can sort of, we'll come on to this in personalized medicine, I guess, as you mentioned, but, if you can earlier in the process be better representing the patients that you might ultimately be treating, or you have a better spectrum of the disease from the get-go in terms of like maybe the number of individuals available in the research, perhaps it will better capture where things might progress through the rest of the pipeline. But many hurdles, and although AI will be very useful, it's not necessarily going to solve all of the problems along the whole pipeline.
Absolutely. Thank you for framing that. I want to come back to the idea of rare diseases since it's been touched on and sort of by all of you, and we are now in Rare Disease Month, the month of February. Rare diseases are defined as those that affect less than 200,000 patients in the United States, but it's actually something of a misnomer that makes them sound a lot less significant than they can be. 70% of them start in children, many are severe or deadly, and taken together, they affect more than 300 million people worldwide, which is not at all rare when you think of it that way. So I wanted to ask, all of you, Leena, maybe you can start, what the sort of unique challenges are, what the treatment opportunities are for these kinds of diseases.
Yeah, like you mentioned, some of them can be so rare. And as I previously mentioned, the patient population is so small that oftentimes patients go misdiagnosed or undiagnosed, and doctors or specialists, aren't really equipped to help guide parents, either because, chances are they've never seen another child has this condition, and or each child presents so differently oftentimes. And so, I think because of the fact that we're so, some of these diseases can be so rare, yet we just sort of get lumped into categories that, make it challenging for pharmaceutical companies or biotechs to really become interested in the disease itself, because of the small patient population. But the fact that there could be overlaps with diseases that affect larger groups of patient populations is something, that is worth kind of looking into and focusing on, because if you're able to sort of effectively treat a smaller disease group, could you then take that therapeutic to a larger patient population and essentially effectively treat that disease as well, and I think that I'm grateful for the work that's being done at NYSCF, and I think that we really should start focusing on those overlaps, because we're not looking for a treatment for just one disease at this point, then we're looking for treatments for multiple diseases.
Absolutely right. And INAD is often referred to as a pediatric form of Parkinson's disease. So certainly there's a lot of commonalities there that we can learn from. Dan and Vic, anything that you'd like to add there?
Well, I think Leena touched on some really important points. I think if we're going to think about the common diseases as being complex, and if we're going to say that one of our great challenges for these diseases is to, quote unquote, convert them into simple diseases, simple single gene or few gene diseases, so that we could subtype our patients, then effectively what we're really saying is that we want to convert complex diseases into a group of rare diseases, right? That's effectively what we're saying. And so in order to develop any kind of treatment, then let's say I subtype Parkinson's into 50 subtypes that are now much rarer than Parkinson's disease umbrella. Well, I need a way now to navigate the treatment of those diseases. How do I navigate what happens then? Let's say I have a gene that I think is working for a subtype of patients for Parkinson's. Well, I might look at neuronal axonal dystrophy as a case example for how this was done for a disease where there was let's say a single gene mutation. So we need to learn the way for complex diseases through navigating simple diseases. And so I think that we've had some great examples. I mean, the astonishing success of treatments for spinal muscular atrophy for children, for a subset of those children, has shown us that there's hope. There's hope for disease modification for these groups of diseases with genetic interventions. If those experiments and that treatment had not been developed, we'd have a lot less hope. So that gives us a way, I think, to chart forward, what we do for more complex diseases. And sometimes, and Leena also mentioned this, sometimes it's going to be just methodology. There's going to be a deep biological relationship. So since we're talking of Parkinson's disease, there's a disease called multiple system atrophy that I'm very passionate about. It's a devastating disease, and it's a disease that is related deeply to Parkinson's. The same protein that aggregates in Parkinson's aggregates in MSA, but it progresses much more quickly. And now many biopharma companies are interested in this disease because they have therapies directed at Parkinson's that they want to apply in a more rapidly progressive disease where there may be advantages, both regulatory advantages and advantages related to the speed of progression of the disease for designing a clinical trial. So I think, just as Leena said, we want to find win-win situations to bring partners to the table where they might be focused on rare diseases, but have larger disease, more common disease indications in mind.
Absolutely. Well, let's move on now to the opportunities for AI to take on these challenges that we've just heard about. We've heard, of course, a lot about AI controlling our phones and the ads that we see and making us buy things on Instagram we don't need. This is an example for me. But why is it now that it's garnering so much excitement for disease research and drug discovery? What can it do that other technologies that we're kind of using as part of the standard pipeline cannot do? Dan, would you like to start?
Yeah, sure. It's again, it's scale as we've been, not just we here at NYSCF, but the field in general have been able to do ever bigger experiments, constantly generating more and more data, and not just generating data and then kind of being done with it and putting it to one side, but remembering you've done it. It just amasses. There's more and more and more data out there, and what it's allowing us to do, whether it's a clinical medical record, whether it's from an experiment, is allowing us the ability to aggregate that data, combine it all together, and look for patterns. And at some level, this is what it comes down to, is looking for patterns in the data to which something meaningful can be applied. And it's simply because it's become accessible and become easy to do this. That is why there's been a shift toward it. Advances in technology like self-driving cars, at some level they've all originated from the same sorts of concepts at some level. And it's great to have collaborated with Google, for example, on this work where we took advantage of things that they've built called neural networks that were trained on images of cats and dogs who knew that one day they would be incredibly useful for a piece of work like this in Parkinson's disease. Again, by hand, by eye, even with a giant team of people, looking at the images in the way we would traditionally look at them is just, we wouldn't have teased this apart, the signal that we're detecting to classify the two individual groups. It's got too big for people to do by hand sometimes. And so being able to bring in tools like machine learning is terrific. At the same time, you have to be very careful with it. It can incur biases and it can incur mistakes if you don't ensure that you sort of set data up correctly and take good care of your data. And it can lead you down paths that might be completely incorrect. So, human intervention has to be there, making sure it sort of checks and balances are in place. But as a tool, it's becoming incredibly powerful to us in a number of different ways.
Right. So garbage in, garbage out. So how do you make sure that you're not putting garbage in when it comes to a study like what you were talking about, Dan?
Again, being incredibly diligent about how you're recording data, so putting it into databases that faithfully capture it, or whether it's even starts at maybe a clinical level, like other medical records being captured correctly at some level. Beyond that, though, it's making sure that you have safety checks for mistakes if they occur, and you can catch them and you can go back and change things if mistakes do occur. But ultimately, it's making sure. Yeah, it's a very good question. It's making sure that the data is captured in as reproducible a way as possible, and that you capture in some ways as much as you can from the get-go. You don't know what small piece of data might be useful at some point down the road. It just has to be kept clean and well organized.
Right. Definitely a lot of effort went into that. And in the study that you talked about, what were some of the ways that you made sure that the cell images that were being generated of these millions and millions of cells were kind of as reproducible and high quality as possible?
Yeah. So in taking, like I said, close to 6 million images, it was all sorts of things. It was from the way a laser was functioning on a microscope, was the intensity of the laser functioning as it should, through to, did we put the right number of cells in a given, what we call a plate? Did we make sure that the signal we were detecting from a different color, maybe, was it the same from batch to batch, experiment to experiment? And we built tools, and we have incredible, data scientists here. We have great engineers that along the way made sure that everything from timings of things happening in our process down to the second happened the same way every time. It's just an enormous amount of care and attention put in across the pipeline, really, to try and minimize artifacts along the way. There are countless things, but they all add up at some level.
Right. So all of this automation that you mentioned is pretty essential for all of that.
Yeah, to do it at the scale that we did, I think at some level it requires it. Human hands are great, but at some level, if I've had my coffee in the morning, I might be great, but if I'm sleepy in the evening, I might start making mistakes. And the hope is that the robots, of course, won't do that, and they'll do the same thing the same way every time.
Right, absolutely. So what about the findings from this study do you think, the most surprising to you?
I think there are two. One, obviously, that it works to detect, can separate Parkinson's patients. And of course, this was done in skin cells, so that is in itself surprising, though there is rationale for why that is the case. I think one of the things that popped out was that we could identify individuals incredibly well. And so at some level, there is a cellular fingerprint of people, if you like, and that was very, very surprising to us. And what was perhaps more remarkable is that in the study we had a handful of samples from individuals that we had been able to collect twice, years apart, and even when collected twice, years apart, we could still tell that they had come from the same individual to begin with. Quite remarkable, quite surprising. But yeah, I think obviously the biggest takeaway was that it could indeed identify Parkinson's patients independently of healthy people, and that in itself was, that was the intention, and that it worked is fantastic.
Yeah, absolutely. Vic, what is your take on this, as someone who is not really involved in the study, but who is, kind of looking to find these sorts of signatures and features of disease that can be targeted for treatment or for progression, for early intervention? What is your reaction to those findings that Dan just outlined?
Well, I think they're really exciting, and they speak to the fact that I'm someone who looks at cells a lot, and there's just going to be, we're going to be very biased about the way we look for cellular phenotypes. Our group was really among the first to identify the first pathologies in stem cells from patients who had Parkinson's disease, familial mutations of Parkinson's disease. This is a decade ago now. And we did painstaking experiments where we took a patient with a mutation, we genetically corrected them, and we found the first pathologies in these cells. But we were biased, right? Our version of doing something unbiased was to use high throughput screens to identify key pathologies related to the proteins that aggregate in these diseases. And every method you use is somewhat biased, but we were also trying to take a very broad approach and not go down one particular path. But in general, as humans, we have biases, and we're going to look for things that we're interested in, for example, and there's only so much we can do. And so the idea that we can capture huge amounts of information that are really beyond our capacity and look for patterns with algorithms is very appealing. So in general, I think this is certainly an approach that we need to add and brings a lot of value to the table. But exactly as Dan said, it doesn't replace the need for constructing the experiment in a way that you're really accounting for all the many things that he just mentioned that could go wrong. And so, one could imagine that you could take fibroblasts from patients who are from any cause, and they could cause differences. So we should be very careful about and ask the next question about how specific is this to Parkinson's versus some other disease. And these are all things that you're all thinking about, but you want to constantly be doing good science with these methods. You just want these methods to be part of your toolbox, but that doesn't mean that data is just going to solve everything. I think this is a really important point. And there are many companies that say, we do AI, we do machine learning. We the data itself is not going to solve our problem. And these methods individually are not going to solve our problem. But when used judiciously and when coupled with really good, scientific clinical acumen, they just increase the capacity of what we can do. So one could think about taking patients, for example, who have the same mutation but very different outcomes, and asking if Dan's method could distinguish between those. So that brings together different worlds. You might talk to clinicians who are very deeply invested in understanding the biology of the disease in their patients and say which patients would be the best to now put through the JAX-NYSCF platform. And bringing those worlds together would be very, very helpful, I think. And that just means that there's, like with any great science, there's lots of other things to do. And but again, I think that we can't assume that just pressing play and watching this unfold with just a whole bunch of data is going to solve the issue. But there's no doubt that the capacity here is really exciting, and we need to do this. So yeah, they're just some general responses. We're obviously thinking about similar methods in looking at genetic data, not just cellular data. But even there, if I just take genomic data and say it's going to subcluster my patients, just having a whole lot of data, I don't think that's going to be the case. But if I look at that data through the right lens, then I might see signals in that data. So the onus is still on us to design a good experiment.
Absolutely. That's a great thing to keep in mind. Speaking of comparison to other diseases, Leena, we've been excited to be working with your foundation over the last while on applying some of these methods to INAD. Can you talk a little bit about that work and what sort of potential you see in it?
Yeah, a little bit. I can let Dan discuss the science, but from what my understanding is that we're able to use this technology, the AI technology, to detect phenotypes in the cells of these patients, which we were not successfully able to do in the past. And so the technology, that's become available has allowed us to see sort of consistent disease phenotype in these cells, and with the ultimate hope of potentially screening existing FDA-approved drugs against these cells to try to get to a point of having therapeutic options available to these children. I think that that's a huge step forward for a disease like this and an organization like ours to get closer and closer to sort of that precision medicine. And hopefully there's some benefit to come out of it. The symptoms, as far as symptoms are concerned, or improvements are concerned, in the way the disease manifests itself.
Absolutely. And yeah, the benefit with the FDA approved drugs is that they've gone through all of this sort of safety testing. And so they, can have a quicker path to the clinic. Dan, anything you'd like to add about the work that we're doing with Leena and INAD Care?
Yeah, I think Leena sort of touched on sort of the big take home messages there. It's obviously, working toward using these approved libraries of drugs to find therapies. I think, if we do, and we're hopeful that we will, it doesn't stop there though, as we spoke about. Even an approved drug would still need a lot more validation. And so the great thing about a collaboration like this with Leena, and that we've done with others as well, is that although, yes, we have the skin cells and we've been using skin cells at the moment, we've also made stem cells. And so, we have this great technology to convert a skin cell into a stem cell. And once it's a stem cell, we can turn it into anything else. And so it could be into a neuron, for example, and obviously maybe one might think even more relevant to a disease like INAD than a skin cell. So if we have a drug or a group of drugs that look promising, it allows us then to do some element of secondary validation in a whole new cell type. We can take the same patients that we've been working with, but instead put the drug onto neurons that have come from those people and see if we see a beneficial effect in one way or another in those cells as well. So I think there's a lot of scope in sort of how we take these projects forward and combine. Again, to Vic's point, it's not this isn't a one and done sort of scenario here. It's a tool in a toolbox, but the toolbox is getting ever bigger, and that's perhaps like the best part of this, is that there are more tools to hopefully get better answers at the end of this.
Absolutely. Well, looking forward from this approach, and so we have this initial proof of concept study, which was, thankfully successful. How do we go forward from this? Do we think that, for example, we might be able to identify Parkinson's disease in skin cells early in progression? How early do we think that might be possible? What implications might there be for intervention strategies, like Vic mentioned, are a really important priority in these diseases?
It's a really good question, and one we have asked ourselves.
One from the audience also.
It's good, terrific question, and one we've asked ourselves numerous times. The problem with those kinds of questions is, to Vic's earlier point, of how patients develop. And so, these have at that point to be what we call longitudinal studies. You would ideally have collected a set of samples much earlier in life, maybe made a prediction on the samples at that point, and hopefully have followed then that back to that people or back to that person to the point of which they are diagnosed with something. But that could be, if you take a skin sample at 30, someone might not develop Parkinson's until 50, 60, so how do you do a study that long becomes the challenge. There are, again, this is why biorepositories are so important. Like if we can, continue to collect material and track it. One perhaps great example of this is in a study we've done with a researcher at Brigham and Women's, Tracy Pierce, who has been working with a cohort of nuns and priests who have indeed donated material over many years, and there we were able to see in human neurons that we grew from stem cells signatures of Alzheimer's, and actually able to correlate that to then tissue, post-mortem tissue as well. So the studies are possible, it just needs to be set up from the get-go, and that perhaps is sometimes the challenge.
Yeah, and I think this relates to some of the questions that I'm seeing in the chat as well, that having these really carefully curated samples, and this is where the partnership with patients becomes so important, where we can't do this without the incredible generosity and partnership of our patients, donating samples, donating their time for these kinds of studies. But I think that it's come up in the chat, can you correlate findings with the brain? So one of the studies that I'm doing that's supported by JAX-NYSCF involves building a fairly large repository of matched brains to iPSC cells, so that we have stem cells matched to brain, where we can correlate. And what we're finding now is we're able to discover new pathologies in the brain based on what we're seeing in the patient stem cells. So that's an interesting effort that I think is important, has come up. To speak to Dan's point, sometimes there are going to be remarkable families that could be helpful. I follow a family in northern Spain, multi-generation, that has an alpha-synuclein mutation, and there we have siblings that have the same mutation that have very different outcomes. Some of them get Parkinson's, some of them do not. It would be really interesting to use the kinds of approaches here for families like this, where they're rare. Again, we were speaking about what's the connection between rare and common diseases. They're rare, but they do get classic Parkinson's disease, but they have these very different outcomes. So would your algorithm be able to predict that? I think those would be really follow-up experiments.
Absolutely. So the potential is sort of very broad in terms of, I guess that's the benefit of the unbiased approach, is that you can see applying it to many different diseases as long as you have patient cells available, which is really exciting. We have just a few minutes left, but I do want to come back to this, this topic of precision medicine and the idea of tailoring treatments to individuals or subgroups of patients. So what are the implications of these kinds of approaches that we've been discussing today for realizing precision medicine? Might there be ways to use AI in a predictive sort of standpoint? For example, there was an audience question about predicting seizures and strokes. What would it sort of take to get us there? Vic, do you want to start with that, and we can bounce it around?
Well, I think that it's really important to have the data upon which we can train these algorithms. So, so the idea of doing careful natural history studies, as boring as they might sound to a biopharma company that wants to just get in there and use a drug.
Could you explain what a natural history study is?
A natural history study is to observe patients over a period of time to collect the kind of data that is saying is so important, to collect biorepository specimens, bioimaging, biometrics. So now we can put sensors on our patients, with Parkinson's disease, and track their movements, not just once every 3 or 4 months, and then track them all the time. We have video-based apps that they can use on their cell phones that can give us a whole bunch of information. So we can now capture data in ways never been able to capture before, both biological data, behavioral data. We need to build these longitudinal data sets, because in the end, I think that if you're going to think about personalized medicine, someone's previous history is the best control for how they respond to a drug. How they have been doing in the last year or two, their slope of progression, the way their brain has been changing or their body has been changing, is the best way of understanding what happens when you give them a treatment and then you give them a placebo. You might see something change again. That's going to be the best way. If you're going to talk about personalized medicine, you need longitudinal tracking of individual patients. And companies so far have not invested the billions of dollars that they've invested in drug discovery. They've not invested in this simple process of gathering longitudinal information from patients. So I would say that if, as a group, we're going to advocate, we should advocate for that kind of data set being built in different diseases of interest, because in the end, that's going to be a treasure trove that we mine, so that these algorithms can be shown to be, to be as useful as their promise is.
Well, that is a hopeful future. Dan, for your final question, I'll actually draw on one that we got from the audience, which is, can you see a future application of taking cells from a patient with Parkinson's, generating a model of their disease, and then screening drugs specifically for them?
Quite possibly. And the reason I say yes, almost, is that we've seen examples of this in not Parkinson's, but in other cell types already. So there's been examples in the literature of using cells to grow heart cells, and you can sort of see them beating in the dish, and there have been studies that have tailored drugs to find the best treatment for individuals. We're also seeing it in the cancer space. That's perhaps like the best one in some regard, where again, we can grow in theory tumors in addition, again, screen drugs against those cells to find which may be best at destroying those cancerous tumors. This is only going to keep growing as a space in of itself. And so the possibility of doing this in Parkinson's, yeah, I think it's entirely a possibility. The hope, though, is maybe that we get to a point where we can generalize better, and that there may be, ways of of doing this at some level that's a combination of perhaps genetics and perhaps longitudinal data that Vic's described, but instead of that being so patient-centric, it's aggregating it across many patients to get these patterns and trends, instead of, you know, it being very generic at the moment with just, you know, you have a diagnosis and so you get this drug. It's shifting away from that. Vic can probably, again, has spoken to that, but that's probably the direction it goes. But I mean, they're the models we're kind of using right now, and we'll continue to do so and try and perfect and keep moving forward.
Thank you. Leena, we'll give you the last word. What do you see as the sort of ideal future of how we would tackle brain diseases like INED, and what do you think it'll take for us to get there?
You know, I think that we're just hopeful, right, that these breakthroughs in science will actually lead to treatment options and cures for these devastating diseases sooner rather than later. And I think that by learning more about like the root cause of these diseases, as well as the overlaps that affect these larger patient populations, you know, we're more hopeful for these treatment options for multiple diseases, not just, you know, one disease at a time. And I think that by collaborating and data sharing, working together to find solutions that will work across these diseases will certainly help us get there a lot quicker. And, you know, kind of clearly working in silos doesn't seem to be as effective, right? So, you know, it's nice to see all this focus on therapies and these brain diseases, and you know, all this kind of efforts kind of going towards different treatment options for diseases like ours that are very devastating and hard to watch, and you know,
yeah,
it's a lot of hope.
Great. Well, thank you for all you've done to tear down some of those silos and really accelerate some of the progress that will be no doubt meaningful for many other patients with these diseases. So I want to thank you all very much for this insightful discussion. And it was just such a blast. I feel like time just flies when in with a group like you guys. So thank you very much. And I want to thank all of our audience participants for your questions and your participation. We tried to get through as many as we could, but we will try to address those we didn't in follow-up. So hope everyone stays well, and to see you at a future NYSCF event. Thank you all so much.
Thank you.
Yeah.